Hello students, welcome back to our Prelims Masterclass wherein we are covering current affairs from the last 1.5 years. I'll be taking care of the science and technology, and yesterday we covered substantial portion. Now today we are getting into the theme called biotechnology. So whatever articles we will discuss today will be related to biotechnology. Biotechnology is very important segment. Yesterday I showed you the trend that UPSC is focusing on biotechnology segment a lot. Almost 2 questions every year we see from the same segment. So there are many articles from this segment. We will deal with some articles today and some articles we'll do tomorrow. Okay. If you have any doubts, you can ask. I'll check the comment box. Just give me a second. Yeah. I'll keep on looking the comment box. If you have any queries, you can ask. So, before we start, there is a notification from Narayana Academy that our 1 year and 2 year courses are active. If you want to take admissions, if you want to check the details, you can go through our website. 1 year prelims cum mains integrated course, 2 year prelims cum mains integrated course. All the details are provided on our website, or you can just click on the link provided in the description box. Okay. Now, let's start with our articles. We'll not waste time. We'll just start with the articles, and the segment is biotechnology. Now see, simply biotechnology is all about using biology as technology, or using biology as a tool to solve some of our problems. So let's say there is a bacteria. In that bacteria, there is an immune system. There is some particular gene. If we want to use that gene in human beings, so we can use that. Or let's say we're using that bacteria to solve our problem. Let's say there is some kind of oil spill which has taken place. So there is oil in the water. You want to remove that oil, or you want to remove some kind of waste. So you can use that bacteria. So you're using biology or biological products, biological processes to solve our problems. That's all. That's all when it comes to biotechnology. Okay. Now, in this segment, the first initiative or first article we are going to discuss is about Decode Initiative. Decode Initiative. Now see what has happened. Why this has been in news? Because we got a news from United Kingdom. United Kingdom has recently completed genome sequencing of 5 lakh people. 5 lakh people from UK. Their genome sequencing has been completed, and this was done under this larger project called Decode Initiative. Now we need to know what is this genome, what is this genome sequencing to understand this news particularly. So let's go for the basic part. What is genome? What is genome sequencing? Now first of all, look at yourself. Your body is made up of cells. There are so many cells. There are billions of cells in your body, right? You look at any other organism also. They are also made up of cells. There are some species which are single celled, like amoeba. Amoeba is made up of just one cell. And you look at human beings, they are made up of billions of cells. Now, in each cell, if you take one single cell, in that cell, we have something called nucleus. This is the nucleus. In that nucleus, we have something called chromosome. Now, it's not like there's only one chromosome. There are so many chromosomes. There are total 46 chromosomes, that too in pairs. So, one chromosome, other chromosome will be in pairs. So, there will be total 23 pairs of chromosomes in human beings. I'm talking about human beings, human cell, one single cell. Okay, we have these chromosomes. Now, if you take this chromosome, just zoom it, you will see that this chromosome is made up of a very long chain or a long segment which we call as DNA. DNA, deoxyribonucleic acid. Now see, this is your DNA which you see, or this is your DNA. It's a very long chain, long structure. Okay. Now this is the part in which all your genetic information is stored. Let's say My eyes, the color of my eyes is black, the color of my hair is black, but there are some people whose hair color is, let's say, white or blue or whatever. Now, how come this is decided by my body whether the color of my eyes or hair would be black or blue or whatever? That will be decided by these DNAs. Or when you look at these DNAs, in this DNA, all this information is already stored. So we can say DNA has all the information or all the information and all the instructions needed by our body. And these instructions which are stored in this DNA, that will decide what will happen in your body. Okay, now these DNAs are present in each single cell. Obviously, in each cell these are present, and the cell will work the way this DNA will say them to do a particular task. Okay, now cell clear, chromosome clear, DNA clear. Now when you look at this DNA, I'm saying DNA is a very long chain. Okay, if you take just one segment of it, one part of it, then that one part would be your gene. So DNA, when you talk about DNA, it has got all the information, all the instructions needed by your body. And here comes just one particular gene, just one part if we take, that is your gene. This one gene will tell one specific characteristic about you, one specific trait about you. So when I gave you this example that my color of my eyes would be black. So I have one gene which would say that the color of my eye would be black. Then there are so many other genes also available in my body. So if I take just one segment, that would be gene. Okay. Now comes the question, what is genome? If you have understood DNA, if you have understood gene, now you can understand what do we mean by genome. Look at the DNA again. This is the entire structure of DNA. If I again zoom it, I'll see a pattern. You see, this is the zoomed version. If you see this zoomed version, you will see that there are some chemicals or there are some things called nucleotides over here. And there is a particular order of these nucleotides in this DNA. So you can think about this thing like, let's say the entire DNA is your book. In this book, everything about you is written. And this book is written with some words, some letters. So these are those letters, this language. This is the language of this DNA. Now you see this language or these words, you'll see a pattern. There will be some nucleotides. There are 4 nucleotides, A, T, C, G. And entire DNA is written in a particular order. Let's say you see over here, there's a particular order over here. Okay. Now comes the point wherein we want to know the exact order. Exact order of these nucleotides. When you get this exact order of these nucleotides in your DNA, we call you have sequenced your genome. So when I say this word genome, genome means the complete set of DNA that you are having. If I get the complete set of DNA from your body, I can understand everything about you. I'll know everything about you. I can understand, is there any genetic disorder in you? I can understand why there's a problem in your genes. What kind of problem is there in your genes? I'll understand everything about you. I'll even understand what kind of disease you might get in future because of particular genes, right? So this complete set of DNA is your genome, and getting the exact order, the exact sequence of this complete set of DNA is called as genome sequencing, which is being done by scientists. Okay, good. Now, genome sequencing, lab process to read DNA based sequence, and this will reveal the genetic information to us. Now, this will be helpful specifically in precision medicine. Precision medicine, what happens is, normally when you go to doctors with some kind of a problem, doctors will give you the same drugs which they are giving to other patients who are suffering from similar kind of problems. But see, it is possible that your genetic structure is different from that person's genetic structure. So we can go for a new method of giving medicines to people on the basis of exact requirement of a person's body. So if doctors know about the genetic setup in your body, then they can give you exact the kind of medicine you need. Even the amount of medicine will also vary for different people, right? For one person, he or she might need eight tablets. You might need 4 tablets only, because there is difference in the genes. So doctors can be very precise when they are giving the medicine. And this is what we call as precision medicine, which is possible because of genome sequencing. Okay. Now, few things, if I have to just jot down what kind of benefits I am going to get because of genome sequencing. First, I can identify rare disorders. Sometimes you come up with some people you observe, they're having some kind of disease which is very much unique or very rare. We don't see such disease in any of other people. There are only very few people who get such disease, and that disease is caused because of some problem in their genes. So let's say when their parents reproduce this baby, when the genes were formed, when this DNA setup was formed, there was some change or some mutation, or there was a kind of faulty gene present in someone's body, because of which this rare disorder or rare disease is coming up. So here, if scientists can understand your genome, they can sequence your genome completely, then they can identify why you're getting this particular disease. They can understand which gene is leading to this disease. And if they can understand, later on, they can come up with a therapy or a solution wherein they can edit your gene, they can change your gene, they can modify or replace a different gene with this gene, this faulty gene, right? So genome sequencing would be very helpful. Even there are some diseases which are not genetic diseases. See, there are some diseases which are genetic. You can't do anything about it. But there are some diseases for which there is a reason in genetics. So sometimes doctors ask you, let's say you go to doctor and say that I'm getting some breathing issues, or let's say I've got asthma or anything. Doctors will ask, is there any person in your relatives or in your let's say your parents or grandparents, someone is having same kind of disease or not? They're checking the genetic element involved in such diseases. So when you know genome, when you know the exact order of your genome, then this is going to reveal the genetic basis of many diseases. So many normal diseases, let's say even cancer, you can understand, is there any basis in genetics when it comes to this cancer? Okay. Then let's say a child who is yet to be born, you can understand whether this child will have any genetic disorder, any problem, that you can understand because you already have the genetic setup with you. You know the entire DNA of this person. Okay. Now this is something we can understand for ourselves. Okay. When it comes to doctors, they will get a lot of help from this genome sequencing. But apart from this, when we are studying science, when we are studying biology, when we are studying evolution, we will get a lot to know from this genome sequencing. You see, COVID-19 came, right? COVID-19, just one microorganism disturbed the entire life of this planet. Now, here comes something called evolution. COVID-19, we saw that it was mutating, it was changing its behavior. There was some change taking place in this virus, right? So, how do viruses evolve? This is something scientists try to study, and when they study the genomics, when they study the genome, they will clearly understand how this mutation is taking place, how viruses are evolving. And it's not just about viruses, even all the species on this planet, how do they have, they are in a position to evolve, how this evolution is still going on, how it has happened till now. Everything will be very much crystal clear with this studies of genome sequencing. Is this basic part clear? Now, if you understand genome sequencing, you see, to do this genome sequencing, there are many projects being run in the entire world. Now in India also we see some projects. For example, first we started this Indigen project. So it was a pilot program. We just started with doing genome sequencing of few people. So we started with 1,000+ people. We took the sample, DNA sample of 1,000 people or more than 1,000, and we started doing this genome sequencing. Later on, government launched a larger program called Genome India project. So here we took 10,000 plus, or 10,000 to be specific, Indian genomes. And this is of different variety. It's not like you've taken DNA from just from one place. Let's say you go to one place in Madhya Pradesh and from there we have taken 10,000 people and their genome. No. We've taken from different varieties, diverse population. Okay. So this is a very good project to understand the species which is living in India, or we can understand human population better by genome sequencing. Okay. And the news, if I go back to the news, I said UK has completed genome sequencing of 5 lakh people, and this was done under a larger initiative called Decode Initiative. Now this Decode Initiative was launched in the year 1996. It actually started in Iceland, and there the idea was to go for genome sequencing of large population. Why large population? Because if you take the genome of just few people, you might not understand how the genome would be, or how people would react for any particular medicine throughout the region, because you've taken few samples only. You need diverse samples, you need more samples to understand the genome, understand the evolution, everything in detail or in a better way. Okay, so this is how this Decode Initiative becomes important. Okay, any doubt? Please ask if you have any doubts. Otherwise, we'll continue with the next article. Anil Gedela, hi sir. Kindly pick current affairs classes only from Hindu and Indian Express. Anil, we are specifically taking from Hindu and Indian Express, but there are some articles which we do not see in Hindu or Indian Express, but they are highly relevant for examination. So I told you yesterday too that we're taking some articles from PIB also, or we go for researching articles from different sources, because we understand that it's not like UPSC is specifically going to ask only from Hindu or only from Indian Express. They look for the news, they look for what is emerging, what is exactly happening. So this awareness part is needed, right? So awareness will not come just from a particular newspaper, and we are preparing for exam, so we will look at what is relevant for exam, right? So obviously anything covered in Hindu and Indian Express, that is something will be something that will remain our primary source to look for. Okay, now moving ahead, a related topic. Now this is how I have structured things, that related topics will be merged at one place. Now there's something called One Day One Genome Initiative, launched by India. So in India we have a Ministry of Science and Technology. Under this ministry we have something called Department of Biotechnology, right? And under this Department of Biotechnology we have an autonomous body called Biotechnology Research and Innovation Council, in short BRIC. Okay, and there's one more agency of BRIC, that is called National Institute of Biomedical Genomics, NIBMG. Now these organizations have coordinated or cooperated to come up with this initiative called One Day One Genome Initiative. What is this initiative? Simply they are saying, we are going to do this genome sequencing, we are going to come up with the genome details every single day about microorganisms in India. See, we are talking about microorganisms. Earlier we talked about human genome project, right? Genome of human beings. Now here we are talking about genome of microorganisms. So you see microorganisms like virus, fungi, etc. These are very important. There are so many microorganisms which are even present in our body. See, if you look at human cells which are present in our body, those are in billions. Let's say 30 billion human cells are present in our body. Then I'll say 39 billion microorganism or microbe cells are present in your body. So our body is also full of many microbes or microorganisms. So you should know about microorganisms. Now here, this initiative is specifically saying that we are going to come up with the information of genome which is related to this microbes or microorganisms every single day. We'll try to come up with information related to at least one microorganism every single day, and this will be published. This will be published free of cost for the researchers in India as well as throughout the world. They can study the microbial genome. Okay, so very good initiative we can say from India side. Just try to remember the agencies involved because that is something important from prelims perspective. Okay, now this I don't think I need to tell you. You can understand what is the role of microorganisms. Still, for the basic part, when you look at microorganisms, they are present almost everywhere. They have a role to play in our life. When you look at our digestive system, our gut. Now, our gut health is not just dependent on our food. It is also depending on the microorganisms present in our gut. So they are playing some role anyway. So they have some role in the ecosystem, in agriculture, in human body, even in diseases. We're getting many diseases because of microorganisms. So considering all these aspects, we are looking at these microbes and we are coming up with research on these microbes. Okay, very easy, very short news item I can say. Yes. Now the next one. What is the news? Genome accordion. Any idea what is accordion? Accordion. So, genome accordion. First, understand what is accordion. Accordion is this device which you can see over here. This is your accordion instrument. You must have seen in any movie or videos, etc. So, what you do with this accordion is you expand it whenever you want to, you contract it whenever you want to. This thing is possible with this device or this instrument called accordion. Now we are talking about something similar to this accordion in some microorganisms. So there are some viruses, specifically the viruses belonging to this pox family, pox viruses. Like you must have heard about smallpox, chicken pox, or this monkey pox, right? So some recent studies have claimed that if you look at these viruses, specifically these viruses, they have a special ability which is very similar to this accordion. In this accordion, what happens? Expansion, contraction, expansion, contraction. Similar kind of thing is observed in these pox viruses. Okay, what they do is they expand sometimes and then they contract also whenever needed for the sake of survival. So when their survival is at stake, let's say some viruses, these smallpox viruses have entered your body. They've entered. Now they are fighting in our body because our body is having some immune system which is fighting with these viruses. Then let's say you have taken some medicine from doctor. That medicine is also trying to kill these viruses. Now in this case, these viruses will use this new tactic which we call as genome accordion. So you see, first what they'll do is they will expand whenever there is a stress on them. Our body's immune system is attacking on them. At that time, they are going to expand. They are going to make more copies of themselves, their DNA. Or if I say, if you look at their genome, they are going to expand their genome, make it longer for the sake of survival. And this is done by making duplicate copies of genes. More genes will be added. Anyway, some genes are being killed by your immune system. So here, gene duplication is happening, and this is how This virus is expanding in size. Interesting. Now, after expansion, second stage all includes mutation and selection, or simply understand this extra gene which is added over here. Earlier this was the genome, now some more genes are added, that's how the genome became more long, and now all these extra genes which are added, they will start mutating, they will start changing themselves, changing in a way to fight with this immune system. So to fight with immune system, if this gene requires to make some changes in itself, it will make those changes. So mutation will happen over here. Now it can fight with immune system very properly. And let's say once all this fight is done, everything is done, now it will contract. How it will contract? Whatever this extra gene that was added over here, now this will be cut and removed from the body. And this is how this virus first expanded and then contracted back. Very similar to this accordion. And that's the reason we are calling this phenomena or calling this mechanism as genomic accordion. Okay, genomic accordion. So now you can understand this news item. Pox viruses use a unique evolution mechanism known as genomic accordion. Okay, this is the recent study. Clear? Good to go. Now, because we are preparing for preliminary examination, so we must certainly know about some facts about this pox family. So we have got smallpox, monkeypox, chickenpox. So you should know these basic facts about these viruses, at least these viruses. So which virus leads to smallpox? So the name is variola virus. Then for monkeypox, it is monkeypox virus. The same name is there. And for chickenpox, you have varicella zoster virus. Now see this particular chart you will get in the form of notes. You can download those notes, or if you want to download this PPT, all this will be provided in the application, Narayana IS Academy application. You can download it, and in the free materials you will get the notes, the complete compilation of the science and technology material, as well as these PPTs also will be provided over there free of cost. Okay, so still it's my job to tell you, so just go through this, I'll say. So these viruses, name of these viruses, try to remember this very clearly. Now as far as the severity is concerned, this the smallpox is very severe kind of disease. This virus affects your body very badly, and this can lead to death of people. Okay, as compared to smallpox, monkeypox is little milder, and as compared to monkeypox, this chickenpox is way mild. Okay, now right now what is the status? As far as smallpox is concerned, it has been eradicated completely in 1980. Okay, as far as monkeypox is concerned, it is endemic in many countries, this is Central and West African countries, and it is still spreading in many parts of the world. And as far as chickenpox is concerned, we don't care much about chickenpox now, because it has become a normal illness for us right now. So we are even getting vaccines for it. So no problem as such. Okay. Now the transmission, usually this transmission of this virus from one person to the other happens because of respiratory droplets or infected lesions. Okay. This would be common over here also. Here additional thing, if you are in contact with some animals, let's say monkeys, so this can happen. Okay, symptoms are more or less the same: fever, headache, etc. And as far as vaccine is concerned, this is something important, I'll say. Okay, because we have anyway eradicated smallpox, so we don't need this vaccine for this smallpox. As far as monkeypox is concerned, it is recommended only for high risk individuals and not everyone. And this chickenpox, we have vaccine available and that can be given, so no issues. Okay, so just try to read this basic factual part, at least revise it once. Okay, done. We're done with third article, I guess, right? Let me check the comment box if we have any doubts. No doubts. Okay, now The next article is related to CRISPR-Cas9. Any idea about CRISPR-Cas9? This was earlier asked in the previous year questions too, and this topic is relevant for examination, considering not just one aspect that this was asked in the previous year questions, but this is one of the important technologies or cutting edge technology in terms of biotechnology. So right now, biotechnology which is leading, which is a sunrise industry right now, this CRISPR-Cas9 plays one of the most important roles in dealing with biotech. Okay, so here the context is CRISPR-Cas9 is transforming medicine with precise gene level therapies. So what is exactly CRISPR-Cas9? First, let's understand the basic part. See, let's say in your body there is a genetic problem. Let's assume that there is a genetic problem in your body. Okay. Now, you want to solve this problem. Earlier, the doctors used to say it's a genetic problem, we can't do anything. But now we have got biotechnology. Using biotechnology, we can solve this problem. How we can solve it? Simply, we will take your DNA, okay, and in that DNA, whichever gene we find as faulty gene, we understand that the problem that you are getting is because of one particular gene. What we will do is we will try to take that particular gene, we can cut that particular gene and add or modify other gene. Let's say I've taken your gene and now I want to cancel it out. Cancel as in, what I will do, simply one gene I will add additionally. Let's say I'll take one gene from a bacteria and add it in your DNA. Or let's say a faulty gene, I'll just cut and remove it. That is also possible. Now, this is all part of gene therapy or whatever genetic changes, genetic modifications we are making in a person. Okay. Now, to do this, we need something. You want to cut something, you need a scissor, right? If you want to stick something, you need a glue. So, such tools are needed. You can't neglect that part. So, an important tool in this biotechnology field is your CRISPR-Cas9. It's a tool, or if I have to simply say, it's a scissor. It's a scissor that we use to cut your gene. Cut your gene. Wherever I want to cut, precisely at that particular location, I'll cut it. Okay. So it's a molecular scissor, we can say. So CRISPR, when we say, actually we take this CRISPR from a bacteria. So there's a specific bacteria in which there's something called CRISPR. So it's a part of its what we call defense mechanism. So what happens, sometimes virus attacks bacteria also. Virus attacks human beings, virus also attacks bacteria. What bacteria do? They will cut that virus. Okay. So to cut, they have something called this CRISPR. Okay. And here this Cas9, when we say, Cas9 is an enzyme which acts as molecular scissor to cut the DNA at specific site. So overall, the name is CRISPR-Cas9. The CRISPR includes special proteins, special enzymes. One of that is Cas9, which is going to cut wherever needed. So where to cut, that will be provided. That information will be provided by the CRISPR. Okay. So this is how we cut the genes. Now, is this technology important? It is very important. Now we are utilizing all this cutting part very much whenever we are going for any particular application in the field of medicine specifically. So let's say we talk about sickle cell anemia disease or this thalassemia. Now, in such diseases, we know that the problem lies in the genes. So we have to solve it in terms of genes. So doctors now have treatments to solve these problems at the genetic level, because now doctors are in a position to use CRISPR-Cas9 technology. Okay. Even for many cancer therapies, now CRISPR-Cas9 is being used, because see, cancer is happening. Why cancer is happening? Because we see the growth of some cells. Some cells are growing, and that growth is not controlled growth. They just keep on growing. That's a problem. So if we can change or modify the gene over there present in that particular cell, then maybe we can stop that particular growth, right? So that's why even in cancer therapies we use it, and it has potential in many infectious disease treatment like malaria, HIV. There also we might come up with some therapies or some solutions by using CRISPR-Cas9 technology. Okay, this is the application in the field of medicine. Then you have the field of agriculture. See, in the agricultural field, nowadays we talk about genetically modified crops, right? What you have done, let's say we talk about BT cotton. Because of BT cotton, our production has increased substantially. What we have done is we have taken a particular gene from a bacteria, Bacillus thuringiensis. There is a bacteria. Not just bacteria exactly, it's not a bacteria, rather it's a species or nematode we may call it. But we have taken a particular gene from a different organism and added it in a plant, that is your cotton. And using this gene, we are killing the insect which was causing problem to the cotton. So there's an insect called bollworm which creates problems for our cotton plant. So now we have added a gene which can kill it. So we are killing it, and that's why BT cotton is very good. Now we're talking about many other GM crops. Now when you look at these genetic modification, to make this genetic modification, we might need to cut a particular gene, and for that we need CRISPR-Cas9. Okay, so if we utilize this technology for agriculture sector, we can increase the crop yield, we can enhance the disease resistance in plants. If there is a particular disease these plants are getting, any insect or anything is infecting these plants, then we can create some resistance in their body. Okay, this is clear. Then obviously there will be some scope in biotechnology itself. So let's say nowadays we talk about biofuels, we are making biofuels. So to make biofuels, we can again make use of this CRISPR-Cas9. You cut a gene, add an extra gene to produce biofuel. So all this can be done, and even for new drug discoveries, if you want to look for new drugs, you have to make some changes in the genes and check whether this drug will work or not. So all these things, application part of CRISPR-Cas9. I hope at least the basic part you remember in one single line. CRISPR-Cas9 is a scissor. It's a scissor which is going to cut your genes. Simple. Okay. Let me check if we have any doubts. Hmm. Kindly explain about metagenomics. Metagenomics is just the study part. Means researchers are studying about the genomics. There they are talking about metagenomics. So wherein in different environments they'll check how things are going to work. So they'll just go for different samples and they'll study it. Okay, nothing so much specific about it. Okay. Any other doubt? No? Okay. So the next topic is, what is prime editing? Now I talked about CRISPR-Cas9 technology, using which we are editing the genes. We were modifying the genes. Genetic modification was taking place. We are cutting the genes. Now the same technology has evolved, and now we call this new technology as prime editing. Okay. So here also we are going to cut, we are going to modify the genes, and this will be done in a precise manner more precise manner as compared to your CRISPR-Cas9 technology. The traditional CRISPR-Cas9 technology, as compared to it, this prime editing will be much more precise. Okay. Now, how it will be done? So there are some technical parts like this prime editing guide. RNA is there. Cas9 is over here. The Cas9 is same as the Cas9 we saw in CRISPR. how we differentiate this prime editing from our CRISPR is because of the element involved over here, which we call as PEG RNA. Now see what happens exactly when we were cutting DNA at a particular location. That means we were cutting a particular gene using CRISPR-Cas9 technology. You look at DNA, it is double stranded, right? DNA is double stranded. So this double stranded, when it was being cut by CRISPR-Cas9, it was cut at their two different localities, or this double stranded part. It was not like only single stranded part was being cut. Both the strands were being cut by the CRISPR-Cas9. So in case of prime editing, we say that it is so precise because of this particular RNA, it is going to guide this Cas9 in such a way that it will just cut where it is needed. So it will just cut the one part and not both parts. And that's the reason we say it is a better version of the CRISPR-Cas9. Okay. So we have got some templates over here, editing templates given by this PEG RNA, which will be helpful over here. And that's how this prime editing will work. Again, if I have to just sum up it in one single line, CRISPR-Cas9 and prime editing both are doing the same thing. That is, they are cutting the gene at a particular location. The difference simply lies in, we can say, let's say I have a paper. So I want to cut somewhere at a particular location in that paper. So I'm just ripping off the entire paper from the middle. Now this is being done by CRISPR-Cas9. But if I just look at what is written on the paper and I just cut it where exactly it is needed, at a particular, let's say on a particular word or a particular letter, there if I do it this much precise, then this is being done by your prime editing. Okay, because we have an RNA which is guiding it to become very specific. Okay. Now an important topic in your biotechnology is this gene therapy. Although we already know about gene therapy, that whatever genetic modification we are doing, that's your gene therapy. Now recently there's a news that India conducted its first human clinical trial. This is the trial of gene therapy for a disease called hemophilia A. There is a disease called hemophilia A. It's a genetic disorder. So we wanted to make changes in the genes, and that we have done. So this therapy is called, this method of medication or solving this problem is called as gene therapy. And this is being done for the first time. So it's an important news, we can say. Now what is hemophilia A? Exactly what happens? Now you must have heard about something called blood clotting. Let's say I got a cut over here. What will happen? Some blood will come out. For some time blood will come out, but within few minutes what will happen? Blood clotting will take place here at this particular location where this cut is there. The cells over there will interact with each other. They'll interact with the neurons, and my brain will give some direction to these cells that you have to ensure that blood clotting takes place over here, that no more blood should go out from my body. Okay, when this blood clotting will take place, after that you won't see any blood coming out, right? Now this is a natural programming done in our body. This is the natural process that happens in our body, blood clotting which happens. If blood clotting will not take place, what will happen? All the blood in your body will start coming out. It will not stop continuously, it will come out. Now what will happen if there is no blood in your body? Whatever oxygen you're taking, that won't reach to any part of your body if there is no blood. It is the job of blood to take this oxygen to every single cell, every single part of your body. And if this oxygen doesn't go there, you can't expect your body to function now. So you will die. To be specific, you will die. Now, when it comes to patients who are suffering from this disease called hemophilia A, they have some problem. This problem is a genetic problem. There is some problem with their genes, wherein this clotting factor number 8 is not present, or is limited, is in a very limited quantity available in their body. Now this clotting factor number 8, this is the same factor which is responsible for blood clotting in our body. So whatever blood clotting is taking place in our body, that is happening because of this clotting factor. And if there is a person in whose body this clotting factor is not present, there is deficiency, then we say this person is suffering from hemophilia A. So it's very obvious. This factor is not there, no blood clotting. No blood clotting means even if this person gets a small cut, all the blood will start coming out and this person might even die. Even the minor injury can cause a big problem for this person. Okay. Now, because this is a genetic problem, now we want to solve it at the genetic level using biotechnology. So, what we use is gene therapy, right? Gene therapy means we are going to make changes in the genes present in your DNA, which is present in your cells, right? So either we will add one gene or we will cut the gene which is causing the problem. Okay. Now, first we can replace the faulty genes. So what we'll do is we will find out whichever is the faulty gene, we'll cut it, we'll remove it, and we will add the good gene over there. Or we can inactivate malfunctioning genes. That means we can cut and remove it, simply done. Or we can introduce new or modified genes. So let's say we are taking a gene from other organism and that also can be added over here. All these things are possible. Now these genes can come from the same body's person or other person also. So if it comes from the patient's own cells, it's called autologous. And if it comes from other donors, it's called allogenic. Although these words are not that important, just for the sake of understanding I'm telling you. Okay. Now when you look at this gene therapy would be coming up in different types. One of that type is germline gene therapy and other type is somatic gene therapy. Now something interesting you need to know about your body. If you look at the cells in your body, there are different kinds of cells. The important cell number 1 would be your somatic cell. Somatic cells, and second is your germ cells. Now see, if you look at your body, most of the cells in your body are your somatic cells. Most of the cells. Let's say I take example of your muscle cell, blood cell, nerve cell. All these are your somatic cells. And there are very few which we call as germ cells. And these are involved in the reproduction process. So your sperm cell or ovum cell. Now sperm and ovum, they play major role in your reproduction process, right? They have the main role to play in the reproduction. So these cells are different. We call them germ cells or gametes, right? We use this term called gametes also. Now, somatic cells and germ cells, both are different. This somatic cell is very individualistic. It's about me, it's about my body. Whatever changes you make in my somatic cell, those are going to affect only me, not my children. Even if I give birth to a child, or let's say my generations continue, there won't be any change in their body because you made any change in my somatic cell. Okay, because it's just about me. If you change my muscles, it won't be like the same kind of muscles will be there in the next child. No, that's not necessary. But when you make changes in the germ cells, that is, if you make change in my sperm cell or the ovum cell of a lady, in that case, the same thing will continue in the next generation. That is, the child that will be born from this person will also have those traits, those characteristics which you have added or changed in that particular gene. Okay, that's why you should know the somatic and germ cell separately. So when we do this gene therapy, where we are doing it, whether we are going for gene therapy on germ cells or somatic cells, that decides the type. So let's say we are going for germline gene therapy. So we are making changes or modifications in your germ cells, in your sperm or ovum or what we call as gametes. When we do this, entire generations, the next generations will also have the same traits. Those will be passed on to the next generation. So simply I can say whatever change I am doing, it's a permanent change. It's a permanent change for the generations, and this raises many ethical questions whether it should be allowed or not, whether it should be restricted or it should be completely allowed, right? This is something which is concerning. Whereas if you do the same with somatic cells, well, these are the cells which are going to affect me only. So whatever you do is not going to change anything in the next generation, right? So ethically it is more or less accepted, governed by many regulations, and that's why it is more or less applicable or is being used in many countries. Okay, but the problem is it's temporary, it's just about me, and I might need to do the same thing with my next generation. If let's say the child born has similar kind of problem, then we have to go for again this somatic gene therapy on that child also. Okay, so this is all about gene therapy. Okay, and recently it was done with respect to hemophilia A. So you should know what is hemophilia A, you should know gene therapy, its types, how it is being done. Okay, any doubt till now? Hello Ram Kumar, any doubts? If you have, please ask. No? Okay, now let's move ahead with the next topic. Now the next topic is your CAR T cell therapy. Very important, and this is in news since last 2 to 3 years continuously. Since last 3 years I am observing this news related to CAR T cell therapy. It's very important, and this has a major role to play in dealing with this disease called cancer, right? We all know what is cancer now, that there is uncontrolled growth of some cells in your body. See, cells grow, that is the natural thing. There was a cell which started growing, and that's why I have become. My hand, which, when I was a child and when I'm right now, if you see the size of my hand, it has changed because the cells have grown. But the cells stopped growing at a particular point. Otherwise, my hand would have been of this size. When cells grow in an uncontrolled manner, that is something we call as cancer. And dealing with cancer is a very problematic stuff, very complicated stuff. We have some therapies like this chemotherapy which is available, some radiation is given. These things are available, but those are very dangerous. When you give the chemo, the people suffering from cancer, you will attack the cancer cells. You will attack those cells using this chemotherapy, but it will also affect the good or healthy cells which are present in your body. So although you're killing cancer cells, you're also killing some good cells which are there in your body. So that's why it's very complicated, right? And even it's very costly, very expensive too. Now we're looking at a new therapy which can solve our problem, which we call as CAR T cell therapy. This is not going to be much expensive as such if you compare it with chemotherapy and all. And most importantly, it is not going to kill your healthy cells. It's not going to kill your healthy cells much. It is way better than the chemo, we can say. Now, what is this CAR T cell therapy? In this CAR T, this T cell is very important. Okay, first we'll understand what is this T cell. You look at your body. In your body, we have blood. In that blood, I talked about this part yesterday too, that blood is composed of many things. You have red blood cells, you have white blood cells, you have platelets, etc. Now, if I just talk about white blood cells, WBCs. Now, these WBCs would be of different types. There would be different cells when it comes to WBCs. Now, if I take just one segment out of it, then that would be your T cell. Okay, T cell is a type of WBC, you can say. Now, this T cell is very famous because this T cell is something which fights for you. Whenever any virus attacks, whenever something wrong is going on in your body, then when we say that our immune system will attack it. So what is exactly attacking? This T cell is actually attacking. So it's part of your immune system, and this T cell is produced in your bone marrow and it matures in a particular gland called thymus. That's the reason it has got the name T cell. T for thymus. Okay. So in simple terms, this is your army. This is your army in your body. This T cell is your army, your immune system part, which is going to attack any foreign particle getting into your body. Okay, if any virus comes, this T cell will attack it. Now, in this CAR T cell therapy, we are trying to make use of these T cells. Anyway, these T cells attack anything that is foreign, anything that is causing problem to us. Now, because the cancer which is there in our body, the cancer cells, they are part of our body anyway. That's why the T cells are not attacking them, because they are part of our body. It is not something foreign. We have not added something from other people or any other organism which has entered in our body. It's part of our body only. So that's the reason T cells are normally not attacking these cancer cells. What scientists will do? They will take this T cell, they will make the modification, they'll go for genetic modification, they'll change the genes in such a way that now this T cell will attack cancer cells. Okay, so same gene editing which we were doing earlier, same we will do, and we will modify this T cell. Now when we modify, we are expressing a receptor called chimeric antigen receptor, or CAR in short. This receptor we are adding over here in this particular T cell. Now using this receptor, your cancer cells will be identified and they will be attacked now. The T cell will identify them as foreign particles or as something that needs to be attacked, and T cells will automatically attack. So what you have done is, you know that you have immune system, you have just tried to tweak it. You have tried to modify it in such a way that we use that immune system against our cancer cells. Okay. So this is all about your CAR T cell therapy. Now, how this exactly happens? First, we take the blood sample of a person. Let's say in my body there is a problem, there's cancer and all going on. So, my blood will be taken, blood sample. From that blood sample, WBCs, specifically the T cells will be extracted. Okay, those will be extracted, and now in the laboratory, people will go for genetic modification in the T cells. So, this modification will happen to produce this CAR T cells now. When this is done, now we will take the same T cells and again put it in the patient, that is inside me, inside my blood, and now the CAR T cells are going to attack cancer cells. This is how this therapy works, and that's the reason we understand we have found a good solution to deal with cancer. Cancer, very important for us to study, and we are dealing with it through this new technology or new therapy. Okay, this is the name of this therapy is CAR T cell therapy. Now, because we have done this in India for the first time, we should know what exactly we have done. First, just look at this current use. FDA has approved its use for blood cancers like leukemia, lymphoma, multiple myeloma. Okay, now India for the first time indigenously has developed its own CAR T cell therapy. See, the name of that therapy is CAR T cell therapy. We have developed our own, and the name of this indigenously developed CAR T cell therapy is NexCAR19. Our own indigenous CAR T cell therapy name is NexCAR19, and it was developed by a company called ImmunoAct. It's actually incubated in IIT Bombay. So in IIT Bombay, they have come up with a small company and they have developed it. And it has got approval from government agency called Central Drugs Standard Control Organization. So this NexCAR19, remember the name NexCAR19, remember the name CAR T cell therapy. Okay. Now, it's not like it's a perfect treatment as such. There is a possibility of some side effects in case of CAR T cell therapy also. Now, because we are utilizing our own immune system to deal with this problem of cancer, there is a possibility that there could be overactive immune response. So if your immune response becomes more than required, excess, in that case, this immune system might attack your healthy cells. So this problem can be there. We call it cytokine release syndrome. If cytokine is being released more, then there can be a problem. And there could be some neurological issues also. People might suffer from some confusion, seizures, some problem with their speech. All these side effects are possible in case of CAR T cell therapy. Clear? Clear with CAR T cell therapy. Okay. Now, the next part. Yeah, do we need any break? The next article that we are going to deal with is related to type 1 diabetes. Okay. And how we are going to make use of biotechnology to deal with this type 1 diabetes. That is an important part. Now, any idea what do we mean by diabetes? Everyone knows diabetes. Everyone must have heard about diabetes. But what exactly happens in your body when you say that you are suffering from diabetes? Can anyone just tell me this basic part? Yes? No idea? Now, see, when we say someone is suffering from diabetes, we say that the sugar in their blood has increased. See, what is this sugar? Whatever we are eating, that is your sugar. So, whatever food we are taking, that is your sugar. And when this sugar enters in your blood and it increases. See, it has, it is supposed to go for generating energy in your body. That the role of that sugar is to help in generating energy in your body, because we humans are dependent on plants for our food or other animals for our food to get this energy, right? Now if you look at our body, we have something called pancreas, right? So if you look at the stomach, if you go the behind part which is behind the stomach, that is your pancreas. Now this pancreas do one thing, they produce a hormone called as insulin. Okay, they produce insulin. Now this insulin is very important. Why it is important? Because whatever sugar that you have eaten or whatever glucose you have eaten or whatever food that you have eaten, that becomes part of your glucose in the blood. Now this glucose or sugar which is present in your blood, it has to be used by the cells. Cells in your body will absorb this glucose and they will use it to convert it into energy. But cells cannot absorb it if there is no insulin. See, I'll repeat. You have this blood, and in this blood we have this sugar, or you can call it cholesterol. And this sugar is needed by cells in your body, all the cells. This has to reach here. But let's say these cells are locked. The lock can be opened only by insulin. So if you do not have insulin, this sugar will not be absorbed by the cells. This sugar will not be absorbed by the cells if you do not have insulin. So that's the reason we say that people who are suffering from diabetes, in their body, either the insulin is not present or their body is not in a position to use that insulin. Okay, so when the pancreas in your body, they are not making the insulin at all, or they're making it in very less quantity, or the body is not using the insulin that is produced in your pancreas. This is the situation which we call as diabetes. Okay, when you say I've got diabetes, that means this is something that is happening in your body. So when this happens, what will happen? The sugar will not be absorbed by the cells. That means the sugar will remain in the blood itself. That means your blood will have lot of sugar content or glucose amount. This is what we call as increased sugar, right? We say that the blood sugar level has increased. Why it has increased? Because this sugar is not absorbed by the cells, because you don't have the insulin, right? Now this diabetes is of different types, and right now what is important for us is this type 1 diabetes. How does a person get this type 1 diabetes? What happens is sometimes your immune system starts attacking the pancreas itself. So there are some cells in your pancreas which are called as beta cells, and these beta cells are actually those cells which are producing insulin. So mistakenly, sometimes your immune system starts attacking these beta cells. They start killing these beta cells which are there in the pancreas. So if beta cells are killed, nobody is going to make this insulin. If beta cells died in this pancreas, then insulin production will not take place in your body. And if there is no insulin, then you become diabetic, right? Your sugar level will increase. And that's the reason you see when any such person gets diabetes or this type 1 diabetes specifically, because in their body, insulin is not being produced at all. There's no cell to produce it. And that's the reason you have to take injections of insulin. Every single day, these people have to take injections. So, tough life, we can say. Now see, we have a news related to type 1 diabetes. Now I think you can imagine what do we mean by type 1 diabetes. So scientists have helped a type 1 diabetes patient produce insulin naturally using stem cells. So somewhat we have tried to make use of biotechnology in such a way that now a person who is already suffering from type 1 diabetes. So type 1 diabetes means no insulin is being produced in the body because in the pancreas those cells, beta cells, which are supposed to make or produce this insulin, they have died. They have died because the immune system has attacked them. And in such person's body, now scientists are in a position to produce this insulin naturally. It's not like we have to give injection every single day. Now you can do it naturally. And how they have done? They have made use of stem cells. Obviously, you need to know stem cells or stem cell therapy to understand how scientists have done this particular thing. So, when we talk about cells, we have a special cell called stem cell. And in simple terms, we call these stem cells as nothing but unspecialized unspecialized cells. Now, to understand it better, just imagine you are born. When you were born, you are of this size. But if we go for that entire gestation period, especially when that reproduction process happened, the sperm cell came, the egg cell came, they merged together, then the zygote was formed. This zygote increased in size, and later on you were born, and you were also increasing your size, and now whatever you are. See one thing, the process started from one single cell. Okay, initially you were just one single cell. From one cell, the cell division happened, multiple cells were formed, different cells were formed. So this process has continued. Now when you look at these cells, some of these cells in the initial part, if you look at these cells, some of these cells are unspecialized, or what we call as stem cells. Now these are those cells which have not yet specialized. See, if there is a cell, let's say muscle cell, or there is a cell, let's say this neurons which we call, right, nerve cells. Nerve cell would be a specialized cell. That cell knows what to do. They have a specific task to do and they will do the same task. Now, it's not like you can teach them to do different tasks. But when we say we have unspecialized cell, that means it has not yet specialized. It has not yet got any specific task. So if you take this cell, you can make anything out of it. You can make muscle cell out of it. You can make nerve cell out of it. You can make anything out of it. Such a cell is your stem cell. So stem cells are cells that can become any other type of cell. So we have stem cell. If you take stem cell, you can make nerve cell out of it. So you can make brain. So the cells which are there in your brains, those nerve cells, can be made from stem cells. You can make your heart cells, you can make muscle cells, intestinal cells, liver cells, anything, anything. But if you take any other cell in my body, let's say today you take this nerve cell from my body, you take it. Now you want this nerve cell to act like liver cell. Let's say I have some problem with the liver, I want to add another cell. So I added liver cell. No, it will not work because nerve cell is a specialized one. Okay? But if you take stem cell, you can make it a liver cell. That is possible. Okay? So these are your stem cells. Now these stem cells would be of different types. One type is called as pluripotent stem cell. Other one is called multipotent stem cell. And the meaning is very simple. Pluripotent means it can turn into any cell. Pluri means plural. It can become any cell, any of these cells. That is pluripotent stem cell. And there are some cells which are multipotent. Multi means only multiple, not all. So there are some limited cells can be made using these cells. Those are multipotent. Now, see this. These are stem cells. Usually we get it from embryo of a child or when you are a baby. At that point of time, we can get it from you. Sometimes we can get some stem cells from adults also, adult people. But because you are an adult, those cells are not pluripotent, they are multipotent. Pluripotent means they can become anything. But pluripotent, multipotent means only few things are possible. But what scientists do in the laboratory, they go for this induced pluripotent stem cell. So they will modify your multipotent stem cell in such a way that it will act as pluripotent. Okay, this is called induced pluripotent stem cell. Okay, now this is called stem cell therapy. Now how it is happening in simple terms, if I have to say. See, I have a stem cell. Okay, I've got diabetes, there is some problem with my pancreas, insulin is not being produced. I want that beta cell which has been completely destroyed by my body. I want those beta cells back, right? But beta cells are specialized cells. They know their task, they do one specific task. But I don't have any extra beta cell to add in my body. If I take it from other people, it is possible that my body will not accept it. So I need beta cell which belongs to my body. Okay, now all these cells in my body right now that I have, they are made from a stem cell. They were earlier unspecialized, they have become specialized. So if I take a stem cell and if I make it, if I convert it into that beta cell which can produce insulin, then my task is done, right? My task is done. That's what scientists are doing when it comes to stem cell therapy. They are taking this stem cell and making it what we call as this beta cell and producing insulin in my body. Now automatically my body will generate or produce insulin. So my diabetes is done. There's no type 1 diabetes now onwards. Okay. This is one method of using this stem cell therapy. There's another thing called islet transplantation. Islet transplantation. Then this is one more method. We are using the gene therapy, we are using the biotechnology to solve this type 1 diabetes. One way would be to make use of islets. What are islets? So if you look at your pancreas, in those pancreas there are special cells called islets, islets of Langerhans. So actually there was a scientist named Langerhans, from him we got this name. So this islet or these cells, these are of two types. One is called alpha cell, one is called beta cell. Alpha cell increases your blood sugar, beta cell reduces your blood sugar, right? And we were talking about the same beta cell till now, that it is actually producing your insulin, right? So instead of, so in the earlier method, what we did, we made beta cells using stem cell. But in this method of islet transplantation, we will directly bring this islet all together, means which includes beta as well as alpha cells. We are going to transplant islet directly in this person's body. Okay, this is one method, which is a good method, we can say. Done. This is how we are dealing with type 1 diabetes. Okay, you should know about type 1 diabetes, you should know about diabetes, the basic part, you should know about insulin, and you should know about this gene therapy that we have available with us now. Okay, done. Now the next part is, or the next article over here is named as biotrig technology. Biotrig technology. Now it's a very simple technology which is being used so that we can deal with the waste part. So let's say in our rural areas we see lot of waste, even in urban areas also we see. Now to deal with this waste material, we are trying to make use of biotechnology. So for this waste management, we have got a technology named as biotech. Now what we exactly do in this biotech? Now, in the last lecture, I remember I told you about this process of pyrolysis, how it was going on. I said that we will go for burning the coal. Last lecture, if you remember, I said we will go for burning the coal without making use of oxygen. Normally, when we burn, we make use of oxygen, but here we will not use oxygen. Without oxygen, we are burning, and the temperature will be very high. So let's say here it will be somewhere around 400 or more than 400 degrees Celsius temperature will be there. It can go even beyond 500, 600 also. Now here, when you do this pyrolysis, because oxygen is not present, the process would be little different. The product will also be very different. Normally when you heat with oxygen, the product that is inside will be burnt. But here no oxygen, so no burning. So what we see is pyrolysis. Now when you do this pyrolysis on any waste, especially we are talking about organic waste, so in the rural part, we get lot of organic waste. Let's say the stubble we have got, or many other stuffs we get. Now, if you collect all this waste and go for this pyrolysis process, it will not be burnt. Rather, we get some new byproducts. One product is bio oil, other one is syngas, other one is biochar. What is bio oil? Well, it's an oil. It's an alternative to the oils that we are using right now. So instead of your what we call crude oils and all, here bio oil can be a good energy source for us. Instead of petrol or diesel, you can use it, or kerosene, you can use it. For cooking oil, you can use it. This bio oil. Syngas. Yesterday I told you about syngas. We discussed the coal gasification part. We said that this syngas is not a single gas but a mixture of many gases which has this hydrogen also, methane also, water vapor also. So this syngas is a very good fuel. It's a very good fuel, right? And then we have biochar. Now this biochar is a kind of fertilizer for us. So this fertilizer can be used in the agriculture very easily. So this biotrick technology is all about going for pyrolysis technology at community level in the villages, wherein we will collect the waste, organic waste, do this pyrolysis process and make these valuable products. So waste to good products. We took the waste material and now we have created something very useful for us: bio oil, syngas and biochar. Okay. So this is about this biotrick technology. It is going to be very beneficial for us. Let's say it is going to reduce our pollution, indoor pollution specifically, right? Then it is going to improve the soil health. When you use this biochar, anyway you're improving the soil health. you're using organic products, you're not using chemical fertilizers as such. When you use that oil is better than using the wood. You're burning the wood, it's better than using that. It generates clean power, let's say the syngas that we're using, and it promotes sustainable rural development, right? This is the basic understanding that we get from the biotrick technology. Okay, so these are the major articles that we were supposed to deal with today. Okay, now biotechnology theme is a bit larger, so the number of articles are also more. So what we will do is we'll deal with some more articles tomorrow. Okay, and we will cover up this theme of biotechnology. Okay, so stay tuned, we'll cover the rest of the articles. Okay, if you have any doubts, please ask. I'll wait for one more minute and then we'll wrap up the session. Yes, any doubts? No? Okay then. So we'll wrap up the session for today. We'll meet in the next session tomorrow. Thank you so much for joining.